Hello, this is my presentation for the submission to Discover Student Research Symposium 2024. My name is Sadid Morshed and I work in Dr. Shah's lab here in UT Arlington. And today I'm going to talk about my research on analysis of polar constituents in mixed plastic, pyrolysis oils using different extraction techniques. So what is pyrolysis oil and why are we interested in producing them? One of the world's biggest concerns right now is how to dispose of plastic waste without affecting the ecological system. Pyrolysis oil is one of the promising results that can help us to recycle plastics, but also we can get fuels that can replace the traditional fuels as byproducts. It can be made from algae, or which is called biopyrolysis oils, or from plastic. Our main focus for this research is to focus on the pyrolysis oils that are made or extracted from plastic by heating them at high temperature without oxygen. This process used to be considered as a form of chemical recycling back in Europe, but as of 2023, they have decided to no longer recycle plastics to make fuels, but instead they are using this process to make new plastics. But here in USA, we can still use this process to make new fuels like gasoline or diesel. But at first, we need to know what is inside this oil so that whenever we can use that, it doesn't harm our environment. So as of now, we do know like when we use or we refine these oils, this produces further or fewer greenhouse gases than traditional fuels. And also it prevents plastics from entering our waterways or microplastics entering into our ecological system. So overall, it is tackling both climate change and microplastic problem that we are having right now, especially because most of the plastic that we end up trying to recycle are actually ending up going to the waterway anyway. So we have these plastic waste which are normally candy wrappers. We took the help of the company named Lumas, and what Lumas is actually doing, they're collecting these candy wrappers and they put this in vessels where these are heated without oxygen, and then we get these oils. All of the oils that we are working with came from their condenser line and which can be further distilled, but the samples that we worked on are actually we actually came directly from the condenser line right after pyrolysis. So we want to know what's inside this oil, and if we have oil, what plastic created those oils. So we're working on what's inside this oil. Specifically, we're going to be looking for at the polar compounds. The reason why we looked at the polar compounds are important because oils tend to be alkanes or olefins, paraffins. These are the things that are actually give the most fuels. So what the polar compounds do in the oil, they either be toxic to the catalyst or they can just reduce the efficiency of the oil. The polar molecules are the one that are containing the heteroatoms like oxygen, nitrogen, or sulfur. What they do is they end up destroying the refinement process. If we know what's inside the oil, we will know the best way to remove them, and we will also know which oil is not going to be that good for refinement. So we have decided to use two different types of instrumentation for this process. The first one is supercritical fluid chromatography. SFC or supercritical fluid chromatography is different than traditional separation techniques because it uses subcritical carbon dioxide as a mobile phase. We say subcritical because a lot of SFC that we're using have modifiers, so when we add a percentage of methanol or ethanol or any type of organic solvent, it is no longer a supercritical state. So instead of the subcritical gas, what it does, it gives us a combination of both the gas phase and liquid phase. So it's extremely fast while being able to solvate a lot of molecules like liquid does. When we are dealing with gas chromatography, it has to be volatile as it cannot pick up non-volatile stuffs while liquid can. SFC can do both while being extremely fast, but SFC can cover everything. So that is why we choose to use GC or gas chromatography. So we have used two detectors for gas chromatography. With the SFC, we only had PDA that only checked the absorbance, but with the GC, we had two detectors. We had VUV, which is called vacuum ultraviolet spectroscopy. That is also like PDA when it just checks the absorbance. We have the MS or mass spectroscopy, which analyzes it and is going to look at the mass data. So UV is relatively new to our field. It only really works with the gas chromatography, and it is a UV in vacuum ultraviolet region. So that is much less or lower than what we have with the SFC. The reason why we have taken this is these carrier gases in GC, like hydrogen or helium that we use, they don't absorb in that region. So in this region, we can have a clearly or we can have a distinct pattern of paraffin, olefin, or alkane or alkene. So we can tell exactly what everything is going by or through looking these spectral data. Now to examine these polar mixtures, the first thing we did, we had all of these different compounds. We ran different mixture, such as oxygen mix, nitrogen mix, sulfur mix, such given in the figure 5, 6, and 7. We looked through the research and we found these compounds are probably going to be in the pyrolysis oils. So we brought the standard of them, we mixed them, we ran them on the SFC, and we saw how they appeared on different columns. When it came to regular pyrolysis oil, the 2-EP and the 2-ethylpyridine column did seem to have the best result among the other one. But when we ran in all of the different columns, we saw which column worked better for the specific compound, like either on nitrogen or either on sulfur. So it gives us a general idea with SFC and which column we should have used. With GC, because of the high temperature, this pyrolysis oil needed to go through, which is around 450 degrees Celsius. We kind of had it a limited amount of columns that we could use. So we choose to use GC1HD, which is kind of similar to RX1, which is a high temperature column. To also isolate the polar compounds, we did 3 extraction techniques. The first one is we use the aerogels that's given on figure 9. We got this aerogel from Dr. Call's research group, so especially thanks to them to get this one. So they had this silica aerogel which has silver on it. So silver is known to grab the olefins, and because it's a silicose, it can also grab some of the polar molecules from the oil. We treated it like an SPE cartridge, stationary phase, and where we pumped solvent through it, and then we put was then what we put it inside that was pyrolysis oil, and we analyzed the aerogel end up picking up from the pyrolysis oil. We also did the liquid-liquid extraction as in figure 10, where we had oil and water, but we saw like they don't mix it generally. So we tried to mix them and we analyzed the water if they ended up picking up something from the oil because water is more polar than pyrolysis oils. We also plan to do water methanol mixture and water IPA mixture in the future research. And then finally, we ended up focusing more on the CBS blades. The blades that we use are suited for mass spectroscopy and they have a stationary phase on top of it. So this stationary phase can end up taking some of the polar molecules from the oil and we dipped it in a conditioning solvent to get the blade ready, absorb all of the molecules. We put it in the pyrolysis oil and then we deabsorbed or deserved in different solvents. That's actually what we ended up doing the most. We can see that different solvents we tried, we ran it on the SFC, we saw that there were some things that came out. Not that much, but still like uh since like SFC is extremely limited because we cannot see any paraffins or we cannot see any olefins. So we ran it on the GC mass spec and we could see that the plain diesel that had a lot of complex mixture. And when you looked at the methanol and isopropanol, which was deserving this from these plates, we did get different stuff. So we haven't been able to look directly to see exactly what we have that's different, but we can see that they produce different chromatograms. So it is extracting something. We are not sure what this is. We also plan to further investigate. We can see that the diesel ends a lot earlier compared to the methanol or and isopropanol, but we end up getting a lot more alkanes later on. We do not know why, but we're trying to figure that out. As we can see, like at like the 30 minutes, we had peaks in the methanol and isopropanol, but not in the diesel. So in the conclusion, we are trying to examine different polar compounds which can have effect on the efficiency of the pyrolysis oil we have extracted from plastics. So we need to further do a research on the CVS blitz and aerogel or the solid extraction process to come to a conclusion. Again, I would like to acknowledge Lumos for providing us with financial resource as well as those pyrolysis oils. And I would like to thank Shimadzu for giving us the instrumentation. If you have any questions, please leave them in the comments. I will try to answer them accordingly.